A lot of mathematics and computer stuff in the lab. And so today we have a whole day statistics. So it's raining outside the shower, and we will use data to shower you today and to give you. But again, in my first talk, I will talk about experimental design. My talk has no mathematics. Don't be afraid of. I leave all the mathematics to other people in other lectures. So. First, I will, talk about experimental design because, the experimental design is critical and important. When you start to conceive a scientific question, try to, have an idea to do it. And the first thing you need to do is experimental design. You have to carefully think about how to do the experiments. So basically today, works. So I will give you a brief introduction. Then I will talk about the whole procedure here. The procedure, there are 5 steps. One is like a hypothesis generation and how to formulate the problem. Then I will focus on the experimental design issues when we try to do this research. And I will briefly mention this. Performing experiments to collect data. This is your job, and you probably know more than me. I will not talk too much on this, and also briefly mention data management, processing, analysis, and also result interpretation and annotation. Because when you talk about the experimental design, these things always relate to data management, data processing, analysis. You cannot separate these things. So that's why I also briefly mention this. Then the rest of the day, different people will talk about all this data analysis. Probably some of the interpretation. In the 5 or 6 lectures today. So I will not talk about details, but I will mention this. And then I will talk about 2 things. One is the kind of like a standard experimental design. And for this one, probably you use this if you have done a lot of experiments. You use this a lot. I just briefly mentioned this, the basic concepts, how to use the principles I talk about here to the standard design. At the same time, I will talk about kind of like complicated experimental design. These things I don't have time to talk about too much, but I will mention this to prepare you. If you have questions about the complicated design, you can at least consult with statisticians, some professionals to do it. Then finally, I will talk about the experimental design for even more complicated design, which is, like I mentioned in the first day, like those kind of system biology or immune modeling experiments, how to design those kind of experiments, because those things, like you need to use differential equation to do the model, or you do this, and how to design those kind of experiments. I will talk about this, briefly also. Again, this experimental design is a whole semester course for statisticians. We usually take this for 1 semester, but I try to shrink this into 1 hour, and but hopefully I can give you some basic concepts, principles how to do this. I will walk through this, to do this. And so for any of the biomedical investigation, here is a procedure. I think it's quite a kind of standard, steps you usually do. And as I mentioned earlier, the first step is you need to, based on the hypothesis, you need to formulate your problem and also have a clear definition or clear statement of the problem. This is actually critical. Sometimes when I work with the experimentalist, this is the problem. I have to go back many, many times to figure out what that investigator wants to do. I will talk about this. Then when you get to the design of experiments, once you know what the problem you want to do, and then you get to the design of experiment, there are several things or principles you have to follow. Otherwise, you finish the experiment, you get your data, probably you cannot get your conclusions. Basically, your conclusions are confounding with something, whatever that you cannot publish. So that's the problem you want to avoid when you try to design experiments. You don't want to waste your money, waste the experimental money, then you cannot publish because of design issues or design problems. And then when you do the here is just performing experiment and do data collection. And this step definitely, or still you need to follow some principles to do it when you really implement your plan and do the experiments. And then after the data collection, then then you need to do like kind of like data management. This data management include like data quality control, you make sure there's no missing, you always have missing data, but how to deal with the missing data or outliers, all those issues, and the processing, some of the data you need many steps until you get one number. I know you do a lot of processing steps until you find one number. Like in particular, like you do a lot of flow cytometry gating. You do a lot of manually doing something, then finally you give some numbers. So you have to carefully doing those things, and also definitely the final data analysis. When you get data, that's your goal. You do the data analysis, get your result, then you do the interpretation, annotation, and publish. So that's the step. And then after this, probably based on this result, the interpretation, you may generate a new hypothesis. Then you go back to formulate another problem, and iteratively do this. This is the process. That's why when you do research, you never finish. This is the cycle. You never finish. So this is the thing. But this is the key steps. And today again, I will focus on the 1 and 2 step and briefly mention the other 3 steps. And also tell you how to link all these steps. Because these first 2 steps is basically more like experimental design. But again, this is closely related to all other steps. So I will, basically talk about this. And the first step, okay, the first step is, the generate hypothesis and also formulate the problem. Basically, I know most of you, when you try to start a problem or try to start a study, you have some ideas, based on the hypothesis. You already know that what kind of scientific questions you want to address, okay, from an experiment. If you want to design an experiment, usually you already have some, question in your mind to do. The question is, when you try to turn that, question sometimes is vague for you, but you need to formulate that question into a very concrete way instead of very general form. You say, I want to study a flu vaccine. What flu vaccine? In what? In mice, in human? This is too general, right? You have to formulate your problem into a very concrete, very detailed way. I will give you an example later. At the same time, when you formulate your problem, you have to consider some kind of key endpoints. Okay, so because you have a question you want to address, so what kind of response variable is closely related or critical to address that question? I mean, this is the thing you have to think about. This response variable. The response variable, I will talk about this later, have to be measurable, or even you can try to select something you can accurately measure, not only just roughly measure something. So you are able to quantify, all these things. You have to think about this, these things. At the same time, also you need to consider other so-called controllable variables or factors or experimental conditions. These things may also, you are interested in, or sometimes these factors have a relation, are related to the response, and also maybe confounding. And you have to think about any other factors, related to your study. So, this is the thing, you have to think about at the beginning of a study. Okay. Then the first thing, okay, again, based on this, some hypothesis, if you want to address a scientific question, for example, here I give you a, example. I want to study whether an adjuvant can enhance a flu vaccine. Let's say H5N1 vaccine response. This is the question. You are interested in. Actually, we designed this study. We are working on the design of this study for somebody, doing this. And so this is a general question, right? Very general question. Adjuvant. Probably in your mind, which adjuvant you want to use. And this H5N1 vaccine probably already have some, some vaccine there in your lab. So you want to do this, But this is the general question you do. Then when you try to formulate that, problem into a detailed question, here are 2 examples. One example, you want to say, the first thing you can state, you can write down the question as, does the H5N1 vaccine with adjuvant work better? Okay, this is the one way. You say that's work better. So this is the one way. The second example, if I say this, is there a difference in cellular biomarker responses measured by flow cytometry, ELISpot, HAI titer, analysis of this anti-H5N1 in primary murine H5N1 vaccination with and without adjuvant? So which one is better? Definitely the second one, right? If you give me the first one, if people ask us, okay, could you help us to do a sample size calculation? Here is a question I want to do. So I say, what? So, does the H5N1 vaccine with adjuvant work better? Could you do a sample size calculation? I say, what to do? How to do it? So this is the too general. You can specify this as a question, your scientific question, but this is not good as a statement of or detailed statement of your problem. You have to formulate this problem into really a question that give you more details. So basically, the second one, basically, why is it good? Because it spells out the problem to be well understood and point the way how to conduct the experiments. So second, this statement, you tell us, how to do the experiment. Not only to tell the problem, but also tell us how to do experiment. Number one, we know we use mice, to do experiment. It's a marine, vaccine with without adjuvant. I already know there are two arms, two groups, two treatments. One is the vaccine without adjuvant, another one is vaccine with adjuvant. So I know these two arms. And also, in this statement, you tell me what kind of endpoint or response variables you want to use to say whether it's better or not better, right? So basically, you want to use, flow cytometry, ELISpot, HAI titer as the, assay to measure the response variable or to measure the or to quantify the response. So this is quite clear. Even this, I can do more details. Even from this still not you can do more details because, like say, flow cytometry to measure what, how to do the gating, ELISpot for what, So you can do more and more. So this is already better than this. Then this is what usually we do. When people give us a problem, ask a statistician to help them to do sample size calculation or to write a statistical section for their grant proposal. Sometimes they come to us with this thing, very simple. Then I have to ask question, then later they give me this. Then I have to go back again or more question, flow cytometry to measure what, to ELISpot to measure what, this titer, at what time to measure, so we have to do this, many cycles iteratively, so until we know exactly what is the quantitative variables we really want to measure, how to do the experiments. So that's not a simple thing. Like I mentioned in the first day, one of our investigators just came to us. Oh, tomorrow my grant is due. Could you do this for today? Yeah, I mean that's really it's not that simple. You cannot just give me this and I just do something. Again, we are statisticians. We are not magicians. We cannot just turn. Oh, here it is. We don't. If you just give this, we have no idea what it is. Even you don't know. You don't have an idea. So sometimes, as a statistician, I have to help the investigators step by step to translate their sentence, English sentence, or the statement of that question into numbers, into statistics, then into a data analysis plan even. Not only how to design, how to do experiment, but also have a plan how to analyze, how to do the analysis at the beginning of the study, the experimental design, not afterwards. Even sometimes even worse is some people finish the study. Oh, here is my data. Could you do this? I find, oh, this data is not good design. I cannot get that conclusion. So that you waste all the money. So that that's the problem usually we encounter. So you have a very well state your problem, very well formulate your problem into doable plan. That's very important. Okay, so this is this is the thing. So that's why for the second one, even still not detailed enough. At least we know the key endpoint, which is the response, also called dependent variable, which is the cellular biomarkers. I know the response at least measured by flow cytometry, ELISpot, HAI titer. So I know what equipment you have in your lab. What assay you will perform on the samples you want to collect. And at the same time, another. See, for this, a good statement of a question also tell me, or not tell me, also yourself, what kind of factors you want to control. And so this is so-called independent variables or covariates. These things, basically for this question is, I know one experimental condition you want to control is the vaccine with and without adjuvant. This, at least I know you want to compare these two groups, in order to figure out whether the adjuvant is better, with adjuvant is better or not. So this is the thing. And so for other factors or experimental conditions, there are a lot of other things. Now I have to sit down with the people to talk about, back and forth, many times. Like, for example, the dose of the vaccine. If you see, let's say you do the two arms. One is this vaccine with adjuvant, another one is vaccine without adjuvant. The question is, what is the dose? So you should use multiple doses, or just do one dose. If the one dose doesn't work, maybe the one dose, very low dose with adjuvant does not help anything, but with a higher dose, maybe. So there's a lot of issues, and whether you do a boost dose at a later day or not, this may also relate to the conclusion of this, and also like the sampling time. When you do, you will do flow cytometry, ELISpot, whatever HAI titer, but after you give the vaccine to the mice, right? This is a mice experiment, I already know. In what day, what time point you will collect those samples to do that? Yeah, different day may be different. It depends on how quick the vaccine response is, or how quick the adjuvant response is. It's at day 5, day 10, what day? So this is another issue we have to think about. Another thing is for the human is simple. You just to get blood. That's all you can get. But for the mice, you can get lung, spleen, blood, draining lymph node, or all different organs. Where what kind of sample you get to measure all those things. So you measure all these things, or you just pick some of this. So there are a lot of issues. How do you control these factors? If maybe some of the effect can be seen in the lung tissue, but not in the lymph node. Maybe in the spleen. Maybe not in the blood. So the question is, you have to. There are a lot of factors you have to think about, and so this is the thing. Then so this is the question. When you try to state your problem or formulate your problem into a concrete way, so that not only yourself, but in particular, if you work with a statistician, the statistician understands how do you quantify, what you will measure, at what time point you will measure things. So this is a good statement of the problem. So that's the thing. I'm not sure you have questions for this. No? Okay. So then after yeah.
It's a very basic one.
Yeah.
So in our lab, we don't have a luxury to interact with the biostatistician at the early stage of our experimental design. So in that case, what is your suggestion for us to go ahead with all the bioinformatics? Because as you said, at the end of our experiment with the data, we just go to the statistician and say, this is our data, please could you do the analysis? That is the usual thing what happens. So we don't have the luxury to have in our team a statistician. So in that case, what would be your suggestion?
Right. That's what I'm trying to promote here today. So do not wait until after data collection, you consult with statistician. At the first day, when you start to think about your problem, start to talk to a statistician. That's a good thing if you have one. If you have a statistician, in your school, where, talk to them at the beginning. When you think, I'm thinking a problem. Even in the first day, as early as possible. This is one thing. Unless you say, I cannot find a statistician here, I don't have anybody to help. Then what you need to do is you need to learn by yourself to do this. You need to come. Yeah, that's why you need to come to our summer school to learn this, right? So when you design your experiment, finally, what you want to do is you want to do the data collection. When you do data collection, then you need to do data analysis. Otherwise, you cannot publish, right? You cannot do anything. So you have to formulate your problem into a quantitative way. That's why I keep talking about you to not just qualitatively or use English to say I want to do this, do that, but question you have in your mind, what's the variable you want to measure.
So to kind of address my question, what we do is we have actually formal data set annotation forms that we actually send to our statisticians, and they have to sign off before they actually the data set's actually created. And so they have their input and they sign off so that they can understand the dependent and variable. And that, I think maybe that was
Right. Okay. So this is the first step. Then after this, then definitely you need to do the design of experiment in more details. In this way, you have to think about some principles. Okay. So when you try to design your experiment in a concrete way, like how many arms, how to do what, do this, how to do it, then you need to think about the three types of variables. When you talk about the variables, some of these variables or factors can be measured. some of these cannot. Even you know that factors, but you don't know how to measure. So, but you have to think about all these three types of things. The first one is so-called independent variable or factors, which is the variable or factor you are interested in, and also at the same time you can manipulate. Like for example, the dose of a vaccine. You can manipulate. You can use 10 whatever, 20 whatever the dose, higher level, lower level. You are interested in that. If you are really I want to see what dose of the vaccine really works. So these things you are interested in, and also you can manipulate it. You can set the level by yourself. So this is good. And sometimes this variable is qualitative, discrete. Sometimes this is continuous. Okay, I mean discrete means the dose levels. I only be able to do 3 levels. 10, 20, 30, whatever. And sometimes it's quantitative. If you want to look at some of the I want to look at the effect of the age. That age may be continuous. Different age. So whatever. So this can be quantitative, can be qualitative, and also these independent variables or factors must have an impact on the dependent or response variables or the endpoints because you are trying to measure. So this is the independent variable. The second one is so-called dependent or response variable. Sometimes also we call study endpoints. This is the really key point is, you want to use this to measure the effect or whatever, that you want to do. So you make sure this variable is easily or efficiently measurable, and also this must be related to your scientific questions you want to address. You want to have a question at the beginning, right? You want to address. But the dependent variable or the study endpoints have to be closely related or the best related to your question. If you have choices, you have to evaluate which one is more related to your question. You collect those data, you really can use those data to address your question. This thing. And the third type of variable you need to think about, so-called nuisance or supplementary variables. Usually this kind of variable is you don't have any interest. Like if you do there's a vaccine study on mice, you are not interested in the age of mice usually, right? I don't care the age of the mice and all the it's 2-week age mice or the 28 weeks of mice. You don't care, but you want to control it. You don't want to vaccine with adjuvant use very young mice, just 1 week age. Then another group vaccine without adjuvant use old mice. You don't want to do that. So that's why even you are not interested, but that thing may be confounding with your conclusion, with your result. So you want to control it. So that's sometimes even may not be measurable. Some of the things you cannot measure. So these things, but you want to control. And even something you may not know. There are many other factors. Even the mice, you don't know genetically how they're different, or maybe you try to use the same genetically same mice, but there are many other potential confounding factors you don't know. Okay, and sometimes it's not easy to control. That's why I will talk about the randomization later. And the basic principle for design of experiment is this so-called max-min control principle. So in brief, usually people call the max-min-con principle. What this principle means is simple. I have 3 variables. I just talked about these 3 independent, dependent, user parameter. There are 3 rules to control those 3 variables. The first one is for the independent variables. For independent variable, you try to maximize the variance or the variation of that, so that you can get a larger effect. What that means? That means if you are interested in the dose of a vaccine, so you try to set up the levels as high range, as large range as possible, or as many dosage levels as possible. That will allow you to easily detect that effect. The range is bigger, that means the effect is bigger, right? If you have dose 0 is the control, for example, then I want to do a dose 10, dose 20, even dose 100, if you can. But if you have enough money, there's no toxicity problem, you don't kill the mice, then that should be okay. But based on all the constraints or conditions, you need to maximize that. So that's give you the more effect, larger effect to detect if you can maximize so-called independent variable. The variable really have an effect on the key endpoints. Okay, this is the first principle. Second principle, which is to minimize the error variance for dependent variable, which is the endpoint you want to measure. Okay. For example, if you are trying to use the HAI titer as the response variable, whether its vaccine response is good or not, you use HAI titer. For that, you have to try to minimize the variance, the measurement variance. I mean the error variance. Now, number one is you need to standardize the procedure. When you do the HAI titer, maybe your lab have 3 technicians. Ideally, you need to use one technician to do it. That's more consistent, reduce the error. Or you may be able to do this to measure this maybe in 3 different labs. Ideally, one lab is better, more consistent, right? So you try to standardize the procedure within the lab, within the use the same person, even try to that's why we call use a reliable individual measure. Do not try to spread out. Do not make this measurement more like a bigger variation. So that's the principle. Also try to use large samples, and also using the homogeneous sample to reduce error variance. This is because when you use, sometimes you do experiment, use the cell line, you buy, the cell lines. Ideally, use the same batch of cell lines you buy. Try to reduce all those, potential variations, because this is the dependent variable. This is the endpoint you really want to do. So this is the second principle, which is minimize. And the third one is the control. So control means, as I mentioned, the last variable is nuisance or supplement variable. These things you are not interested in, but this may have a potential confounding effect to your result. So that's why you want to control. One thing is you want to eliminate the nuisance variable. How do we eliminate? One is fix it as constant. For like as I mentioned earlier, if the age of the mice is not interest to you, so you want to use the same age. You don't want to spread out to different ages. So try to fix that that thing. And also the second principle is use randomization to control the effect of nuisance variables, even for those variables you don't know. So if they have no control of the age, for the mice, I have a bunch of mice and they have this mice is 1 week old. That one is 2 weeks. This is 3 weeks, right? I have to use all this. I don't have choice. In this way, you can try to randomize these different ages into different arms, like vaccine with adjuvant, vaccine without adjuvant. Do not use like with one arm just use younger one, another arm use older one. You try to randomize that to cancel out the effect. You don't want that age play a role there. You don't cancel out the effect. So this is how to do the randomization. And also, if sometimes you can build the nuisance variable into the research design. So if you don't want to do that, for example, I cannot randomize the age. So what you can do? You try to take down the age of each mice, then consider that age as another variable in your analysis. Try to use the analysis step. Get rid of this. So this is how to build this variable into the research design. Make that as an additional factor in the analysis. You can put the age as the effect, then try to detect whether age really is the effect. Even you are not interested in, but at least you can get rid of this confounding effect from the age. So this is the basic principle when you think about from the design, or even during the experiments when you do this, implement this, try to keep in mind all these principles. Okay, so this is the thing, and then I will give you an example using this vaccine study. Again, I already talked about most of this. First, it's maximization. Okay, maximization means set the level of interest to maximize the effect of interest. So a larger range or more levels of controllable independent variable is better. So this is again the first principle. That's why I just mentioned that for the HIV vaccine study, if the dose of the vaccine is one of their interests. So again, use a larger range or more levels, but you have to consider tolerability. You don't want to kill the mice, otherwise you don't get anything, right? So under those conditions, try to maximize that. So this is the easy idea. Minimize, also I talked about all these things. I don't want to repeat this. So basically, try to minimize the error variance for the dependent response variable. As I mentioned earlier, for this vaccine study, you want to use flow cytometry, maybe ELISpot, other things to measure the response variable. So that's you can keep consistent, like use the same flow cytometer, do not try to, today I use this machine. If you are luxury, you have more flow cytometers. Today I use this one, tomorrow I use another one. So that, give you a problem. Try to use the same one, and maybe same batch of reagents, whatever, keep the same experimental conditions, use the same type of mice, whatever, these things, try to, I just mentioned earlier, I don't want to repeat this. So this is the way, try to minimize the error variance of response variable or dependent variable, okay? And the third one, again, also talk about is the control or fix the nuisance variables throughout the experiment. Again, for this vaccine study, I also mentioned that maybe use the same age or even same weight of mice if you can, and consider age or weight mice as a control factor in design. This is the way I just talked about. So this is some of the principle you may need to follow when you try to design the experiments. Okay. Then randomization. There are a lot of things you cannot control. So ideally, when you have multiple subjects, to do a study, try to randomize those things in different arms, in different groups, so that you can, average out the effect of those variables, factors you cannot control, those nuisance variables. Okay, so because there are many things you don't know, even you cannot do it. That's why sometimes when we do randomization, even you do blind studies or single blind or double blind, or when you do human studies, I'm not sure you do clinical studies. I did a lot of clinical trials before, and help people to design clinical trials. And when you do the clinical trials, you don't want the doctors to know this dose is a placebo or is a treatment. You don't want that patient to know what is it. You have to completely blind them, So somebody have to know that. Later we will, open that thing, to know. But in this way, you get rid of the biased effect. Sometimes the doctor have bias, patient have bias. Patients say, "Oh, I did placebo. I just threw it away." There's some placebo effect. So you want to get rid of those things. This is really to randomization. And for this H5N1 vaccine study, I also mentioned this. You try to randomize the mice for the two arms. One arm is the vaccine without adjuvant, which is a control. Another arm is the vaccine with adjuvant. So you try to randomize these mice into these two groups. And maybe sometimes you want, one lab probably is not enough to do all these experiments. You may want to recruit 5 labs or 4 labs to do the samples like flow cytometry, whatever the assay. And then try to, if you can, try to randomize the samples into different labs. Do not just try to ask this lab or your lab. Your lab just do HIV vaccine without adjuvant. Your lab just do vaccine with adjuvant. Then these two arms have a confounding effect with these two labs. You don't know what their technician in the lab is doing. So that's why ideally you should randomize these two arms randomly to do not tell them which you are handling the control group or the treatment group, adjuvant group. So that's the ideal case to use randomization to get rid of uncontrollable factors. So this is the thing. I don't have much time, but I will do. And another important question you always ask is the sample size. People always say, "Oh, could you help us to do the sample size calculation?" Okay, how many mice I need? If you do clinical study, how many subjects, people I need for the study? Because that's the cost. That's really, in particular in the human study, the number of subjects you need. It's a lot of money, And so another thing, how many data points?
Not only the money, the availability. We don't
Yeah. You may not have so many patients, right, in one hospital.
Especially controlled patients. We don't
Right.
get it. Like controlled sample, it's very difficult to get it.
Right. You have to pay them, and that's what we will do. And how many data points or replicates per subject? And at what time, at what location you measure? So that's a lot of questions. And for the sample size, I will not show you how to do sample size calculation. Probably next lecture, Hulin will talk about this. But at least in order to calculate the sample size, you need to give us the information like the effect size you want to detect. You want to compare these two groups. For example, I want to compare this HIV flu vaccine with adjuvant, without adjuvant. The HAI titers. I want to use this as the response to calculate the sample size. Now, at least you have to tell me, what the effect size between these two groups? What's the difference, you think? Probably usually you think, I don't know, otherwise I don't need to do this study, right? This is a chicken egg problem. But I have to ask you, you guess, is 10% difference, 20%? You know, if you say, I have no idea, I hope it's 10% or 50%, then I can do the sample size calculation for you for 10% difference, 20% difference, 30%, I can do all these things. But the question is, you have to guess which one you want to do. You want to detect, use this study, you know, so that's the thing. And also you need to do variation of the endpoint. HAI titer, how big, what's the standard deviation or variance for that? You know, based on your experience, probably you have something. You know, before I did a lot of studies, I already measured the HAI titer for other studies, not this one, but I roughly know the standard deviation is this from my lab. So this thing you have to give us. Definitely, usually the power, you know, you need to. Usually people use 80% power for clinical trials or for standard things. You know, we fix this at 80%, and the significance level also usually people fix at 0.05. You know, why is this? You know, nobody know, but you know that's the value people accept. Use the 80% power, 0.05. You can publish your paper, so there's no problem. So I can guarantee that. So then we can use the software. Basically, there's a lot of sample size calculation software to do this. You know, give you the sample size, how many subjects you need. You know, so all these things. You know, so this is just to do this. Usually, for the simple experiments, simple design, like just compare these two groups. This is easy to do. But for other complicated things, sometimes it's not easy, even there's no software to do. In that case, you probably need statistician to help you to do something. But even sometimes for us, it's out of our control. We don't know how to do it. They're so complicated. There are a lot of things unknown. So but usually when we do this sample size calculation, we use a very simple endpoint response variable, and we make make a lot of assumptions, simplify things, like at least even you don't know how big the difference is, but you have to guess, so this is the assumption, right? So even sometimes the variation of the endpoint, whatever your assay, you don't know sometimes. If you have new equipment, you don't know. Again, then you have to guess. Yes, question?
Can you just expand a little bit more on the variation of the endpoint? As you mentioned, the IgG response, would that be the IgG response of the animal normally at the baseline, the variation?
No, this variation I'm talking about is the measurement variance. Okay. So your assay, your basically your whatever whatever you use to measure those. If I give you measure these things 10 times, you measure how high I am. You use a ruler to do it, right? You do 10 times how big that variation is. But ruler is, depends on what ruler, right? Or use a scale to measure my weight. You do 10 times how big the variation is. That thing. It's not the design. Okay. And another thing is, sometimes again there's no prior information. You have no idea what sample size is. What those you don't know the variation. You don't know the effect size. In this thing, what I usually do is I ask the investigator how much money you have. If you say that, like in the hospital, you do clinical trial, how many patients you have? This kind of patients, how many patients you see a year? So this means I try to figure out their capability, how big it is. Then I just tell them use the rule as large as possible, because you have no idea. You have no idea what effect size it is. So you don't have no idea what you can get. So this is usually based on their capability. Then I tell them, I do this back. Tell them. Okay, if you have this capability, you get this number of patients, then what the effect size you can detect? 10% or 5% or even 20%. Do you think this is clinically significant? If you think yes, it's okay. If I say, only effect I can detect is the 80% difference. This 80% is useless. Then you don't do it. So that's the thing. Okay, usually do. And also the design of experiments at this step. Also, you need to formulate your problem into statistical framework. Not only you talk about how to do it, how many groups, those kind of sample size, and you have to have a statistical framework. Otherwise, how do you calculate the sample size? How do you do the experiment in this way, in that way? So usually form this into a statistical framework, mostly probably the hypothesis testing, basically compare groups or compare several groups, multiple groups, whatever. Kind of ANOVA testing, maybe you are trying to look at several factors, not only one. And also maybe you look at the correlations of two variables, or you try to predict something using regression model. So this is all the standard kind of statistical models or statistical framework. So you have to, usually we have help the investigators to formulate their problem into statistical framework, then to go back to do the sample size calculation, then to really finalize the design. So you need to think about this at the beginning, not afterwards, not after data collection. Oh, could you do a survival for us? Or could you do a regression model? Or the NOVA test? So this should be done at the beginning. You need to have a plan for this. Okay, and that's the step 2. And then the other steps I will be brief. I don't have much time. And step 3 is to perform experiment to collect your data. So this thing, just keep in mind these 3 principles. 1, perform randomization if you can, and also keep consistency. Whatever you do, you try to, because you are trying to reduce the error of the dependent variable. Keep consistency doing things, and keep in mind of this max mini control principle during the implementation of the experiments. Okay, do not forget that. Okay, sometimes I do this that way. So you have to keep in mind all these principles, during. And afterwards, definitely you need to have again I mentioned earlier, when you get the data, you need to have a data management plan. This is good if you can centralize your data into a relational database with quality control and management. So this is the cost definitely. You probably, if you don't know how to do this, you need to ask somebody to do this. But this is good for you, because when you centralize your data, maybe sometimes you want to go back to relook at your data. This is good, easy for you to retrieve your data, get your data. Most of the labs, when they are big, they have postdocs, technicians to do all these things. Sometimes, postdoc put his data in the laptop, and the technician put the data in the desktop. Sometimes even the lab PI, lab director cannot find where the data. So even after 2 or 3 years, you want to go back to do it. Even he finds this data, or he or she finds data, there's any documentation, no protocol. What that means. It just gives a bunch of numbers. It's useless. So centralized, well-designed centralized database will help you to not only manage the data or store the data, but also manage all those protocols. All the standard procedures, so that when you go back, you can still understand the data and reuse the data, even share the data with others. And so this is a good. I always push people. Suggest people to do. And definitely you need to have a data processing analysis plan. You need to have a plan at the beginning, not afterwards, not after data collection. At the beginning, before you do data collection. And definitely after you are done, you need to do, perform data processing analysis. This is based on the experimental design and assumptions, because you already have a good design there. You have already some statistical assumption. You have a statistical framework. You have to use those things to perform data processing analysis. Then finally, you need to get a data analysis report. So have a nice report to finish the result. Finish the analysis. Then you can try to use the result to publish. This is the thing. For analysis, again, I will not talk about analysis in my talk, but in the next 4 or 5 lectures, we will talk about all this analysis. For the analysis, there are 3 different types of analysis. First one is very simple. You can do it by yourself. Basically, do some descriptive statistics, even some plots and graphics to look at the data. What you have outliers, or try to figure out what it looks like. This is just a very simple thing. You can do it. And then the second one is kind of a standard data analysis. Or statistical inference, like a testing, ANOVA test, correlation, regression, whatever, those standard things. You can use any statistical software to do it. Some of you probably know how to do it. Some of you probably still need help, but it's okay. And the third level is advanced modeling data analysis. Probably you need help with, you need a professional statistician to help with this. If your data too complex, complicated, too many levels, like I mentioned in the first day, you don't know how to handle, then, this is the thing you need to consult with statistician and modelers even. And actually, if you design your experiments have this kind of things, that means your experiment design pretty novel. Probably you will get something really, really novel. Okay, so unless you say if you do simple thing, you can get something important. But if you cannot do this, but if you design your experiment that need this advanced modeling analysis skills, that means you really create very unique data or complex data. Okay. And finally, the result interpretation and annotation. At this step, sometimes also you need work with statisticians, unless you probably know a lot of statistics, because some of the result is not that easy to interpret. They need the statistical knowledge, and definitely you need to draft the papers to publish. Then again, to iterate all these steps. All these 5 steps you need iterate. Once you finish one project, you generate a new hypothesis, and design a new experiment, and then redo this. So this is the cycle of this. So I only have 10 minutes to finish more details. Any questions before I continue this? No? Okay. So I don't have much mathematics, just principles. Hopefully you can. No. And so one thing is, let me get into a little bit more about the experimental design. This is a kind of like a standard experimental design. Probably you use, many times probably you want to do is you just do the compare 2 or more groups, like actually in that vaccine study design. I want to compare vaccine without adjuvant with adjuvant. It's just 2 groups. This thing is just, if you, basically you want to, if you only have a single factor, you want to use one response variable, one variable, or single variable as your endpoint. For example, I just look at HAI titer. I don't care anything else to compare vaccine without adjuvant, vaccine with adjuvant. Just 2 groups. HAI titer, nothing else. This is very simple, right? Then you just design this experiment, but probably you need to do randomization, as I mentioned earlier, when you have 2 groups, and then the simple, the statistical model is a simple one-way ANOVA. Maybe sometimes you want to do HAI titer for not only 2 groups, but more groups. For example, vaccine without adjuvant, vaccine dose 1 with adjuvant. Those 2, you want to set up several, level of doses for the vaccine. That's fine too. So this is basically kind of like a one-way ANOVA. Compare multiple groups or just 2 groups. That's the same thing, right? And sometimes you may need to do the randomized block design to control other factors, if necessary. And for example, if you do this multiple dose, like control dose 1, dose 2, whatever, at the same time you want to, there are 3 labs involved in this study. You want to randomize this, do some randomization to get rid of the lab effect I just mentioned. There's 2 labs. You don't want to one lab just do dose 1, another lab do dose 2, another lab do control. You don't want to do this. You want to randomize this. So this is the thing you do. Basically the one-way ANOVA, if you have multiple doses, like dose 1 maybe is a control 0, dose 2, dose 3, dose 4, then you do experiment, you get the numbers for each group, then you just compare them. This is simple. As I mentioned earlier, if you have 4 labs to do this, you don't want this lab 1 to do all dose 1, lab 2 do dose 2. So that's why I put lab 1, you need to randomize dose 1, 2, 3, 4 to all these labs. Make sure to each lab have all the doses, to do this. So this is a simple randomized block design with 4 labs. And sorry for that. And there's another kind of design, so-called multiple factor design, which is the factorial design. This is like 2-way ANOVA, for example. And also there's a Latin square design, so when you have more factors. And this is an example for 4x4 Latin square design. This kind of design also try to get rid of the effect of like the batches of reagents and also the lab effects. If you have 4 labs and you buy 4 different batches for the experiments, and the question is how to assign the dose 0, 1, 2. Sorry, this should be 4. No, this should be 3. 1, 2, 3, whatever. Then you need to randomize this to make sure each of these labs have all this A, B, C, D, and each of the batch also have A, B, C, D, so that make it balanced, so that you can cancel out the batch effect and also cancel out the lab effect. This is the type of so-called Latin square design to get rid of the confounding factors of batch and lab. If you have 2 factors to do. If you have more, if I want to control for the type of mice, and or maybe 4 factors, more and more factors. That thing is so-called full factorial design for more factors. For example, I have 7 factors to control. Then your design is more and more complicated. You know, that when you have 7 factors, if even each factor only have 2 levels, then you need 128 groups or runs. So that's complicated. That's why there's another thing so-called fractional factorial design, try to reduce this number. So that's, you know, there are different kind of designs. I don't have time to talk about in details. You know, again, there's a lot of standard textbooks talk about this. And but when you have more and more complicated designs, ideally you consult with a professional statistician if you don't know how to do this. But you know, for all these things, there's a lot of you know, standard things you can use. There's also some templates you can use to design your study so that you can get rid of the confounding factors and make sure all the confounding factors are well controlled so that there's no confounding. You know, you can publish, nobody question your results. So that's, you know, that's the thing. And for more complicated experimental design, for example, there's something you want to do, some kind of regression, use a regression model to do predictions, or maybe some generalized regression, or maybe some of the endpoint you have the survival data like time to event, or you evaluate like time to death or something. You know, this is more like a survival type of things. And also you have longitudinal studies. Longitudinal means you have multiple subjects. For each subject you measure something repeatedly, multiple times. So this is the kind of longitudinal studies. Also some, you know, right now, the new technologies allow you to do those high throughput bioinformatics studies. That thing is even more complicated how to design it, because, you know, if you use the microarray or RNA-seq, you may be looking at, you know, 20,000 genes. You know, there's 20,000 variables means. How do you do that? And also the immune modeling differential equation model. I will talk about this a little bit later. You know, if you have time about this, probably I don't have much time. And so for all these, complicated design, same thing. You have to keep all these basic max mini control principles. There is all this applies to the complicated design. For the sample size calculation, so this is the more complicated. Is probably you may not be able to get a software to do it. So this you may need statistician to help. Sometimes I use computer simulation to do this, to explore the feasibility and the power. You just do the simple, computer simulations. Tomorrow there's a whole day lecture. Catherine will talk about the agent-based model simulations, and you can do those things. Also, you can use different equation model to do simulations. To explore all those things. And I just I use you 5 more minutes because we delayed for 5 minutes, and to talk about how to design experiment for the immune modeling and also this kind of system biology experiments. Again, I just have time to talk about the principles and probably give you some example, but let's go it. So for this use system biology approach to design, I just copied the slides I did for the first day. And again, for system biology or this immune modeling experiments, we are trying to study the interactions between components of a biological system, and also we need to look at the integration instead of reduction, perspective, and also we need to measure multiple components simultaneously instead of separately, and also we may need use new techniques such as those high throughput omics technologies to do it. So this is the thing, and also when you look at this high throughput data at multiple levels, at the genetic protein and cellular levels, organ levels, even population levels, this is very complicated, and also the relationship may be complicated. The factors, different variables may have a nonlinear relationship. Their relationship may be complicated, and also dynamics. There's a time factor. We are looking at, and also really need support of interdisciplinary quantitative sciences. Not only statisticians, probably you need bioinformaticians, mathematicians, different kind of people to help with this. And for example, for this vaccine study, if we want to do this study as the system biology experiments or do modeling, so what do we need to do? We need to measure cellular level functions. Maybe use ELISpot flow cytometry to measure all the cellular level things, and we may want to measure cytokine, chemicals, and other protein expressions, like use ELISA, Luminex, Proteomics, all these things to measure protein levels. And also we may want to measure the genetic levels like microarray, RNA-seq, whatever, to measure gene expressions. This is at all different levels. You meet up, if you have enough money, do this. This is good and fun. And so at the same time, not only measure one time point. If you do the vaccine, give the vaccine to the mice, then you want to measure all these things at day 0, day 1, day 2, up to 10 days, maybe more days. Usually the vaccine, and depends on how long the vaccine response is. So you want to measure these multiple time points. So that's more. And how to link all this data together? We usually use different equations. You know how to do this, right? From yesterday. So if you link all this data into different equations, how to do it is quite complicated, right? And how to design this kind of study? How many time points you need? How many mice you need? This is a very complicated question. You cannot find the software to do the sample size calculation for this. So usually we do computer simulation. We have a software, D-Discover. You can use other software to do simulations to make sure your design is feasible and also is doable, and you really can get what you want. So this is usually the computer simulation is a tool to help with the experimental design for this. So this is the thing we usually do. And actually for these things, when you measure all these multiple things at different levels for each time points, and they also definitely you need to have a plan how to do the analysis, how to link this, how to integrate multiple levels data into one framework so that you really get a good conclusions, and then you can publish. So this is the thing. And so here this is my last slide. And so in summary, when I talk about the experimental design, so number one rule is do not separate experimental design and data analysis. You do not think, I don't need to think about the data analysis in the design stage. These two steps always linked. Otherwise, if you don't think about the data analysis, you don't think about the numbers at the design stage, you may collect some data that may not be able to address your question. So that's that's the thing you have to be careful. And check your model assumptions. You want to do the experimental design. There's a lot of assumptions about the statistical models, about whatever the experimental conditions. You have to check all those assumptions are valid when you do analysis. When you do the design. The question is how to make your design efficient and cost effectiveness. You don't want to waste money to recruit too many subjects or to kill too many mice, and at the same time you don't want to waste your money to say I get the data but not enough, I cannot publish. So how do you balance this? This is really art. It's not that easy to do, but you have to deal with that. And how to make your design novel? So this is the question. If you just simply compare two groups, everybody know that. People think, oh, what's novel? I know this. Compare this vaccine with adjuvant, without adjuvant. I know this. This is an important question. Everybody know that. I just design this, do this, collect out some things and compare them. Do a very simple thing. If that thing nobody has done it, it's probably you can publish, but otherwise it's difficult. So how do you make your design novel? That's the critical. Make your design novel. That means your data will be novel, your conclusion will be novel, then easy to publish. Probably in terms of the grant application, probably that will be better. And again, this is another critical rule: is consult, collaborate with professional statisticians at early stage of experimental design instead of afterwards. Hopefully in the first day you start to think about your experiment. You should consult with statistician. So that's all I have. Any question? So.